figure shows the saltatory conductions. So there's a myelin sheath or the insulators that are made from the Schwann cells here. If you look at the arrow, the red arrow here is the jumping of the action potential or jumping of the impulse from one nodes of Ranvier to another nodes of Ranvier. So now take a look at the nodes of Ranvier here. The nodes of Ranvier is exposed to the extracellular fluid. There's a concentration of the ions, the negative and the positive charge ions at the nodes of Ranvier. So it means that they are sodium voltage gated, sodium voltage gated and potassium voltage gated channels in here. So it means that they're going to be a movement, depolarization. If depolarization, the sodium channel will open and sodium will move from outside the cell into the cell, causes inside the cell to be more positive, and it leads to depolarization. If it's depolarized, it can jump or it can initiate another nodes of Ranvier to depolarize. Okay, look at the arrow, the blue arrow here. Okay, so this is the myelin sheath. It cannot travel backward. Remember, it cannot travel backward. Always travel forward. Why it travel forward? Because the previous region undergoes repolarization process. Repolarization means the potassium channel is opened and the potassium ion diffuse out and inside the cells more negative. And there's inactivation of the sodium channel as well. So that's why you cannot, the impulse or action potential cannot travel backward, but instead it always travel forward. Okay, so this is the saltatory conduction. The saltatory conduction is faster when you compare with the continuous conduction, because in saltatory conduction, the action potential jump from one node of Ranvier to another. But in the continuous, of course, the action potential just move continuously throughout the axon. It does not jump.